The circadian clock governs nearly every physiological function in mammals, orchestrating sleep-wake cycles, metabolism, and hormone release in a precise 24-hour cycle. At the core of this biological rhythm lies a sophisticated molecular feedback loop driven by the transcriptional-translational regulation of key clock proteins, notably period, also known as PER, and cryptochrome, or CRY. These proteins, in conjunction with their regulatory partners, shape the oscillatory dynamics that maintain synchrony between the internal clock and external environmental cues, particularly light-dark cycles. At the core of this biological rhythm lies a sophisticated molecular feedback loop driven by the transcriptional-translational regulation of key clock proteins, notably period, or PER, and cryptochrome, known as CRY. These proteins, in conjunction with their regulatory partners, shape the oscillatory dynamics that maintain synchrony between the internal clock and external environmental cues, particularly light-dark cycles. Beyond their transcriptional regulation, PER and CRY proteins undergo extensive post-translational modifications, or PTMs, which play a pivotal role in modulating their stability, localization, and interaction dynamics. These modifications, including phosphorylation, ubiquitination, acetylation, sumoylation, and O-GlcNAcylation, critically determine the functionality of the circadian machinery. The intricate interplay between PTMs and the circadian proteins fine-tunes the feedback loops governing rhythmic gene expression, ultimately dictating the precision and robustness of circadian rhythms. Understanding these modifications offers profound insights into the mechanisms underlying circadian misalignment, which has been implicated in metabolic disorders, sleep disturbances, and neurodegenerative diseases. The mammalian circadian clock is composed of interlocking transcriptional-translational feedback loops, or TTFLs. At the heart of this system, the core circadian genes CLOCK and BML1 form a heterodimeric transcriptional complex that drives the expression of PER, which includes PER1, PER2, and PER3, and CRY, which includes CRY1 and CRY2 genes. Once translated, PER and CRY proteins accumulate in the cytoplasm and form complexes that translocate into the nucleus to inhibit their own transcription by suppressing CLOCK-BML1 activity, thereby closing the negative feedback loop. The dynamics of PER and CRY protein accumulation, nuclear translocation, and degradation are not merely dictated by transcription and translation rates, but are heavily influenced by a variety of PTMs. These modifications regulate the stability of the proteins, their interactions with other clock components, and their clearance from the cell. By precisely controlling the rate of degradation and nuclear retention of PER and CRY proteins, PTMs ensure the smooth progression of circadian oscillations. In mammals, the disruption of these finely tuned PTMs can lead to profound physiological consequences, ranging from sleep disorders to metabolic dysfunction. Among all PTMs, phosphorylation is the most extensively studied modification influencing PER and CRY proteins. Protein kinases and phosphatases fine-tune the timing of circadian oscillations by regulating the stability and cellular localization of these proteins. Casein kinase 1, a crucial circadian regulator, has been shown to phosphorylate both PER and CRY proteins, dictating their degradation and subcellular localization. Phosphorylation of PER proteins serves as a critical determinant of circadian period length. In particular, CK1D and CK1 phosphorylate multiple sites on PER2, leading to its degradation via the ubiquitin proteasome system. Mutations in the phosphorylation sites of PER2, such as the well-documented S662G mutation, are associated with familial advanced sleep phase syndrome, a condition in which individuals experience significantly shortened circadian cycles, leading to early sleep onset and wake-up times. A landmark study by To et al. In 2001 identified this PER2 S662G mutation in humans, revealing how altered phosphorylation can drastically change circadian timing. The mutation leads to the destabilization of PER2 and its premature degradation, effectively shortening the circadian period. Studies in transgenic mice harboring the same mutation confirmed that PER phosphorylation influences clock speed, linking kinase activity to sleep phase disorders. While PER proteins predominantly serve as a scaffold for circadian complexes, CRY proteins function as potent inhibitors of clock BMAL1 activity. The phosphorylation of CRY proteins by AMPK, or AMP-activated protein kinase, has been shown to regulate their stability and nuclear retention. Activation of AMPK leads to CRY1 phosphorylation, triggering its degradation and thereby altering feedback inhibition within the circadian loop. The regulation of CRY protein stability by phosphorylation has significant metabolic implications. Research by Lamia et al. In 2009 demonstrated that AMPK-mediated phosphorylation of CRY1 links circadian rhythms to metabolic homeostasis, suggesting that disruptions in CRY phosphorylation may contribute to metabolic diseases such as diabetes. This evidence underscores the vital role of PTMs in maintaining the intricate balance between metabolic function and circadian regulation. Ubiquitination is another key PTM that dictates the half-life of PER and CRY proteins, ensuring proper turnover to maintain rhythmic oscillations. The degradation of PER and CRY proteins is primarily mediated by SCF, or SKP1-F box E3 ubiquitin ligase complexes, which recognize phosphorylated substrates and target them for proteasomal degradation. The β-TrCP or beta-transducin repeat-containing protein E3 ligase complex selectively ubiquitinates phosphorylated PER proteins, marking them for proteasomal degradation. Studies have shown that β-TrCP deficient mice exhibit circadian period elongation due to the prolonged stability of PER proteins. This highlights the importance of regulated PER degradation in maintaining a proper 24-hour cycle. Unlike PER, CRY proteins are targeted by two functionally opposing F-box proteins, FBXL3 and FBXL21. FBXL3 promotes the degradation of CRY proteins, ensuring the timely release of clock BMAL1 repression, whereas FBXL21 stabilizes CRY proteins under certain conditions to modulate circadian period length. Mutations in FBXL3 lead to a lengthened circadian period due to CRY accumulation, while FBXL21 deficiency results in CRY destabilization and a shortened circadian period. Acetylation is another critical modification that influences the nuclear localization and transcriptional repression activity of PER and CRY proteins. Histone acetyltransferase, or HAT enzymes, such as Clock, can acetylate PER and CRY proteins, affecting their interactions with other circadian regulators. Acetylation of PER2 by Clock has been shown to regulate its stability and nuclear retention. Mutant mice with disrupted PER2 acetylation sites exhibit circadian misalignment, underscoring the role of this modification in maintaining robust oscillations. Acetylation may also serve as a signal for subsequent ubiquitination, linking these PTMs in a sequential regulatory cascade. CRY1 acetylation modulates its ability to repress clock BMAL1 transcriptional activity. Studies have suggested that deacetylation of CRY1 by SIRT1 or SIR2-1 promotes its degradation, connecting circadian rhythms to cellular energy status. SIRT1, a NAD+ dependent deacetylase, plays a critical role in linking metabolic pathways with circadian oscillations, making CRY acetylation an essential regulatory node. While phosphorylation, ubiquitination, and acetylation are well characterized, sumoylation or small ubiquitin-like modifier conjugation and O-GlcNAcylation or O-N-acetylglucosaminylation have recently emerged as additional PTMs influencing PER and CRY function. Sumoylation enhances PER2 stability by preventing its degradation, whereas O-GlcNAcylation has been linked to nutrient sensing and circadian regulation. Understanding PTMs of PER and CRY proteins has profound implications for human health. Disruptions in PTM-regulated circadian rhythms are associated with sleep disorders, metabolic diseases, neurodegenerative conditions, and even cancer. Chronic circadian misalignment, such as that seen in shift workers, has been linked to increased risks of obesity, type 2 diabetes, and cardiovascular disease. highlighting the importance of maintaining proper PTM regulation. The post-translational modifications of PER and CRY proteins form an intricate regulatory network that fine-tunes the mammalian circadian clock. Through phosphorylation, ubiquitination, acetylation, sumoylation, and other modifications, these proteins achieve precise rhythmic oscillations essential for synchronizing physiological processes with the external environment. The continued exploration of PTMs in circadian biology holds promise for developing therapeutic strategies to combat circadian-related disorders, ultimately enhancing human health and well-being.